Welcome to the Computational Science Colloquium series. Today I have the pleasure to welcome Arun, if I get your name.
Sethuraman. Yeah.
Who is a new faculty in biology and a member of the Computational Science faculty and the center. And he's going to talk about algorithms and data science applications in biology.
Yeah. Well, thank you so much for having me. Are they able to hear me on Zoom?
Yes.
Okay, they should. Okay, good. Awesome. Okay, so thanks for having me. I mean, it's always fun to meet folks now that we've all kind of not met folks in the last couple of years now. So, would love to, especially if you're a student and, new faculty and whatnot, I'd love to chat with you afterwards, which hopefully we'll have a chance to. My name is Arun. I'm a new faculty here in biology. I was an assistant professor for the last 5 years now at SDSU San Marcos up in North County before I moved here. Trying to get my lab set up and everything this semester, but also trying to connect and recruit new students. So I'll tell a little bit about my research as well as some potential opportunities for folks in my lab. I'm going to tell you a little story about some recent work that I've been doing in developing new algorithms for estimating genetic ancestry and evolutionary history from large-scale population genomic data, which is sort of at the core of what I do. I like to call myself an evolutionary computational biologist. My training is in computer science originally, so I have an undergrad degree in computer science and a PhD in bioinformatics. But my applied field happens to be this field of big data science, which is called population genomics. And what that refers to is if you have data from what is called a population. So I'm going to start with some basic info about what exactly do I mean when I say a population. A population is a nice definition that I kind of collated from a lot of different sources. I like to call it a sort of time-space continuum. So it's existing individuals over time, but it's also existing individuals over space. Freely mating individuals, so these are individuals that can mate with each other from one species, that are statistically less distinguishable. Individuals that might be in the same point in time and space might be more similar to each other genetically, whereas individuals that are far away from each other or exist over different generations might be different from each other. So it's a very general definition of what exactly a population is. So, some examples of population. Here's a nice group of population we all know of humans. This actually happens to be in my hometown in Chennai, India. Bunch of individuals of the same species, in this case, geographically and spatially and temporally existing in the same location. Or it could also refer to, here's a simple population, in this case, shown by this nice little square of individual corals who happen, or sponges in this case, who only exchange genes, in this case, with individuals that are very close to each other physically, right? These are sedentary creatures that don't get to move around as much, okay, at least within this area. Or in a lot of cases, it could also be, populations of sort of semi-clonal individuals like bees. Bees, for instance, have a single queen who produces or reproduces lots and lots of clonally produced offspring who form a part of her colony. Okay? And so they're all very genetically identical to each other within a particular colony. Or in some cases, unfortunately, it's literally one individual. In this case, this happens to be the last existing known individual of a tortoise species on the Galapagos Islands, which then eventually died, and then that entire species eventually became extinct, right? So really, if you think about it, a population has a wide variety of definitions, or at least examples of what exactly it means for individuals in a population. Okay. So then I'm going to tell you about what is population structure. So here's a nice, very sort of classical definition by Sewell Wright from 1950. He says it's a division of a population into completely isolated small strains within which there are individuals who randomly mate with each other. So what that means is that if you consider let's say a population of humans, this world as a population of humans, individuals within one small area, let's say here within San Diego, tend to be more genetically similar to each other compared to, say, individuals in one small town in China, because they are geographically separate. Does that make sense? So we call that as structured populations. So populations of the same species that exist in this sort of continuum. Okay. So here's, like I said, the cartoon example of what exactly that means. So here's a population that's all, let's say that these dots here happen to represent individuals of this population. And let's say that in this case, right, just statistically speaking, all of these individuals, reds and blues, seem to be more similarly or similar to each other, so they seem to cluster around this one subpopulation. Similarly, there's more black and green and reds in this subpopulation. There's more greens and yellows in that subpopulation. So to a statistician or a mathematician, this looks like a cluster. Okay. So here are some other definitions, often very confused, actually, unfortunately. Like Dobzhansky, for instance, calls it a race. You'll see Darwin and other classical geneticists also refer to these as race. Whereas now we know race is actually a social construct, it's not a genetic construct. Okay, so not to be confused with this. So here's a newer one from Slatkin and Nielsen. Says the population is not randomly mating because of, say, geographic structure, right? So geographically, a population of an oak tree that exists on one side of a mountain range, another subpopulation exists on the other side of the mountain range. Okay, why is this important? So, gonna get a little technical here, but I want to point you to some very simplistic example from your whatever biology class that you might have had maybe in high school. Okay, so here's an example of how inbreeding, or we call it non-random mating, of individuals within the same geographical area. Let's say that in this case you have a snake here who happens to have what's called a genotype, or a combination of their genes happen to be a capital A and a small a. And this one snake happens to be a capital A, capital A. Okay, so these two are the same, those two are different. Okay. We call these as heterozygotes, they're different. And these are called homozygotes, they're the same. Now when these two mate with each other, now they're going to produce essentially 3 types, or in this case only 2 types of offspring. Because they, remember, when sexually reproducing species have 1/2 of their genetic material inherited from the mother, 1/2 from the father. Okay. So in this case, if you do the combinations here, you could either have this capital A go with this capital A to produce a capital A, or you could have a small a go with this capital A to form a what's called a heterozygote individual. There's only really, if you think about it, just 2 options in this case. So when you have continued inbreeding, which means inbreeding just refers to the fact that you have individuals of the same type mate with each other. So here, capital A small a mates with a capital A small a. You are bound to produce a small a individual. And let's say that a combination of the small a makes it bad or less fit in this case. If you proceed to do the same kind of mating in this population over time, it's going to increase the number of little a individuals over time, just by this repeated process. And the problem, though, we call this as deleterious mutations. Let's say that it reduces the fitness of this individual. Let's say this snake is not able to produce as many eggs. So that means that individual is less fit. So eventually, this population is going to crash and die and go extinct. We call this as inbreeding depression. So depression in the population as a result of continued inbreeding. And this happens in structured populations, if you think about it. So harken back to, this is sort of traditional, the apple doesn't fall too far from the tree, right? So if you think about it, it's an apple tree, drops its apple. Now the apple that's falling contains seeds that are genetically similar to the father tree or mother tree. So now the new tree that's born out of it will be genetically similar. And if this process continues over thousands of years, all apple trees in that area are all going to be genetically similar to each other. Okay. Now, let's say that one of those apple trees happens to have a mutation that makes it bad, produce crappy apples. So that means all the trees in that area will also produce crappy apples, which would mean that the entire population will crash. This is a real problem, and it's a very relevant thing when you think about human health and things like that. Okay, so for instance, when you think about humans, human populations, here's a really nice paper from 2000 that wanted to see, are really human populations also structured by geographical distances? As it turns out, they are, and I'll show you guys several of these kinds of figures. And it's a very simple stacked bar plot. On the x-axis are individuals. The y-axis is a simple proportion. We call it ancestry proportion. And so let me explain what that means. In this case, if I assume that these are all individuals from across the world, so we've got Africa, Europe, the Middle East, Central, South Asia, East Asia, and then Oceania, and then America. Okay. So from left to right, it's starting from Africa all the way into America. What if I assume that all individuals, in this case all humans, from across the world, are derived statistically from two populations or two subpopulations? In this case, a purple population and an orange population. Nothing biological here, right? This is just a statistical separation, clustering. If I do that, and I separate them based on their genetic composition, here's what I see. I see that majority of Africa and Europe and the Middle East tend to have predominantly orange ancestry, right? Because they are predominantly orange. Similarly, if you look at America, East Asia, Oceania, they have predominantly purple ancestry. And then a handful of these populations right in the middle have mixed ancestry. So they have some orange and some purple. Okay, that's how I interpret this. Any folks think of why they would have mixed ancestry? Any thoughts? So here's South Central Asia. Conquest. Well, conquest and just mating between populations, that's all. Yeah. Okay. And that's exactly in lines with what we know about human evolutionary history. Humans as a species evolved out of Africa. And here's a nice track of when humans evolved about 200,000 or more years ago. And then they left Africa around 70,000 years ago to establish populations across the world. Cool. So the story I'm going to tell you, the example I'm going to tell you, is a much more centered story here. Specifically, I study a lot of human evolution out of Africa using large-scale genomic data from native African peoples. And so specifically, I'm going to tell you a story about 4 of these native African peoples. The Hadza and the Sandawe are populations in Tanzania on the eastern coast of Africa. And the Yoruba and Baka populations are populations present on the western coast of Africa. Okay? And Africa is a very large continent. These countries and sort of geographical areas are separated by large geographical barriers, huge river systems, large forests. And we know from anthropology studies that these populations have never exchanged genes with each other, which means that they've never kind of crossed all of those rivers and moved across to mate with each other. Okay. So what we were interested in is to understand their evolutionary history as well as their population structure. So, big questions. Can we actually detect population structure? How do we do that using genetic data? How many populations are there amongst these populations that we sampled? How do we estimate when they diverged from each other? When did they move away from each other? And how did they exchange genes from each other? Now, all of this given their genetic data. Cool. And so there are several models by which we can actually study this. And these are sort of simplistic models of human evolution. And here are some examples here. Under a very simplistic model, I assume all these 4 populations are present at some point in time and they exchange genes between each other. Or you could have sort of what's called isolation by distance. The farther 2 populations are away from each other, they are more different from each other. Or you could have what's called a source sink model, where one single population kind of gave rise to all other populations and so on. Here are some other ones. Okay? And I'll tell you a little bit about some more of this model in a second. Or actually this model right here. It's called the isolation with migration. So this tool that I'm going to tell you about is a software that I developed called Multiplus, or more so a statistical method really, which infers population structure given large-scale genetic data. So here's a sort of cartoon idea of how this model essentially works. I'm going to start with something called a mixture model. And here's a mixture model. So what I'm assuming here is that if I sample a bunch of individuals in this case, and let's say every individual, their genes are a combination of 2 copies, one from mom, one from dad. Okay. In this case, let's say there's a red one and a blue one. So what this model assumes is that all of this red and blue together can be exclusively derived from one subpopulation. That's it. So in other words, if I take any one individual that is red and blue, they have to exist within one population. They can't come from another population. Now we relax that assumption. And what that means is that in this case, I can say that the red can come from one population, the blue can come from another population. Okay, so there are 2 sorts of models by which you could actually assign this. Both of these come from the same population, or each one comes from a different population. Okay. So here's how we characterize this. Don't get scared by the math. I'll explain what's going on here. So let's assume that if X here happens to be the genetic data that I obtained from all of these individuals, and I call this the multi-locus genotype matrix. That's the data that I collect. Okay. And the parameters that I'm estimating from here are the 2 things that I talked about. The first one is this admixture proportion. That, remember the y-axis on that graph, right? Which tells you what is the probability or proportion of an individual's genotype or genetic material that is derived from one population or one subpopulation. And the second one happens to be some way of characterizing how many populations there actually exist. Okay, in this case, I'm going to call that parameter as K, which is just the total number of clusters that are present in my data. Now, if that's true, then I can write a simple likelihood equation under this, what we call admixture model. So the likelihood in this case of the parameters given the data can be written as, in this case, a simple equation here, which is just the product across all of the individuals, all the genetic material that I have observed across all the reds and blues. In this case, this simple product right here of the mixture proportions and these frequencies right here. There are several other tools that actually have been developed to do this method or to apply this algorithm. Several of these, starting with this method called structure. There's some relatively new ones. When I was doing my PhD, this came out called admixture, which all kind of implement various methods to solve that equation right here. I developed a new one called Multiplus, which is much faster. And what it does is it actually employs a variety of optimization algorithms to solve that likelihood equation. Okay. Again, not going into too many details here. But there are a bunch of different optimizations that I implemented as part of this. It cuts down the total number of iterations quite a lot, which allows us to analyze large genomic data, which means I have thousands and thousands of genes from across thousands of individuals, and the algorithm runs much, much faster. I'm also working with a student right now on parallelizing it using OpenMD. So there's a couple of cool things happening with this algorithm, and I wanted to test to see, how this actually works. And so here's the data that I used. So I used data from this big publicly available data set called the 1000 Genomes Project, which has whole genomes from thousands of human individuals from across the world. And when I say whole genomes, to put it into a sort of computational perspective, each human genome, if you think about it as a text file, each one of those files is about 3.5 gigabases large, or gigabytes. Right? I always confuse between gigabases and gigabytes. So human genomes are comprised of just A's and C's and G's and T's. So if you think of it as a giant text file of A, C, G, T, that file essentially is about 3.5 gigabytes large, each one of those. And here we're looking about, this project alone has about 4,000 genomes out there. So you can do the math there to think about the scale of big data we're talking about in this case. Right? So clearly we need large-scale computational optimized methods to actually get at this data, to be able to do it in our lifetime. And you also need resources to be able to run these kinds of methods as well. So what I did was in this case, I took 10 sets of 10,000 genes essentially across the genome. And I ran it for both benchmarking as well as to estimate structure, specifically from those 4 populations that I talked about, the Hadza, Sandawe, both from Tanzania, and the Yoruba and Baka, which are both populations on the western coast, so Niger and Nigeria. Oops. Here's what I looked at, right? So this is just some stuff that I ran relatively recently, hence the table, so don't freak out too much. So let me just point you to, I compared it with a very popular method called structure, which actually happens to be the most popular, most cited software in genetics. If you look at the time to convert, and the likelihood in this case doesn't even reach close to convergence in any of these cases. And this was, I kind of timed them out after about 15, 20 minutes, essentially. Whereas I same data set across all of these different optimization algorithms that I implemented, they converge much, much faster, sometimes often within 30 seconds, which is pretty darn good when you think about the scale of these sorts of data sets that we are comparing. So pretty good. And this is all assuming that in this case, there are 2 subpopulations from across those 4 sampled populations. This is assuming there are 3 populations across.
What does EM stand for?
EM, sorry?
Yeah,
oh, EM here is expectation maximization. So this is an EM algorithm, and then the other ones are all sort of accelerated EM algorithms. Yep. Cool. So then here's what we found across these populations. So if I assume that there are 2 populations, and here again, just to point you to what's going on here, the y-axis here is ancestry proportion, or the probability or proportion of that individual's genome that comes from either, in this case, a blue or a red population. Again, nothing to do with biology here, everything to do with statistics, right? So this is just a statistical classification. If I assume that, the Hadza seem to be predominantly showing up as a predominantly red population. All the other populations, Sandawe, Yoruba, and Baka, seem to stand out as a blue population, which in itself is interesting because that's what it's showing, is that the Hadza peoples are more genetically sort of unique compared to, in this case, all of the other peoples across Africa. If I further split it, if I increase the number of sample subpopulations, the same kind of pattern stands out. You see that the Hadza people stand out as predominantly green ancestry. The Sandawe people then stand out as predominantly red ancestry. And then the Yoruba people predominantly stand out as a blue ancestry, whereas the Baka people seem to be mixed between the 2, between blue and red, which is these 2 populations, right? And then if you go further down, they all kind of stand out as their own populations. But there seems to be lots and lots of mixture between these populations, which in itself is a sort of conundrum, because remember I mentioned these populations are known to anthropologists as to have never moved around and exchanged genes with each other. So we were wondering what the heck is going on, and I will tell you that what's going on at, maybe later, or maybe at a beer or something. We actually did publish some, actually there's some part of it in my story, where we did actually figure out why the heck is there signatures of gene flow between these populations when we know that there's no physical gene flow between them. Cool. So where do we go from here? I do a lot of sort of programming and sort of optimization algorithm development as part of my work. We're building a lot of extensions specifically for missing data. I recently got an NSF Career Award focused on this idea of how do we implement missing data into the estimation process. And when I mention missing data, genetic data is rife, or any kind of data really is rife with missing data. So these are just, let's say that at one particular location, a particular gene, the algorithm that determines if it's red or blue can't do it effectively. So what does it do? It just basically throws it out and can't effectively call it a red or a blue gene. Instead, it just calls it as an N, which is just unknown or just missing data in this case. And on an average, across all of these data sets, there's about 30% or more of missing data from across these genomes. That's pretty crazy, which basically means you're throwing out 30% of your data or more, right, when you're actually doing these kinds of analysis. So what I'm developing now are methods, statistical methods, to be able to account for the presence of missing data while estimating population structure from these genetic data, which is pretty cool. I'm also working on, like I said, I'm parallelizing this method as well. So it's already pretty fast, but I'm parallelizing it. This algorithm just happens to be embarrassingly parallel, which means you can just kind of distribute the jobs across different processors and then collate everything into one. So it should be pretty straightforward. So I have an undergrad who's working on that right now. So we're doing some pretty cool stuff with that. We're also doing lots of test cases, specifically since I have collaborators who've given me access to large-scale human genomic data sets that are not publicly available. So we're doing what's called chromosome painting. So across all modern populations across the world, we're essentially identifying genetic ancestry across the genome. So it's a pretty fun project. So that's kind of in the pipeline for the next 5 years now in my lab. Okay, so far so good. Any questions so far? I'll tell you, second part of the story. It's totally about how do we estimate structure. I'm going to tell you about how do we estimate structure, but in a much more temporal time point. And so this was a method that I developed as part of my postdoc. And so in other words, can we determine what's called demographic history? If I look at this kind of tree here are chimpanzees, and here are humans. Can I actually build some sort of a tree like this to determine at what time did humans, say, in this case, diverge from chimps? What time did let's say in this case, Hadza and Sandawe diverge from each other? Did they exchange genes with each other, etc.? And this goes back to sort of this model that I kind of alluded to earlier. It's called the isolation with migration model. The model is very straightforward, simple idea of evolution. So here, let me point you to what's going on. Let's assume that this is past all the way at the bottom, and these are present day individuals at the top. So past to present time. Let's say that we all were, at some point in time, we all shared a common ancestor. So which means that, let's say that this was that common ancestral population at some point in time in the past. At some point in time t, let's say they split off into two populations. A bunch of individuals went somewhere else, another bunch went somewhere else. Or let's say there was a big flood, formed a big river in between these two populations. This population went that way, the other one went the other way. When that happened is characterized by this parameter t right here. Let's say after that split, they became their own populations all the way up until present day. And let's say it ends here are the sizes of these populations, how many individuals are present. And let's say that they continue to exchange genes between them, which means, let's say at every generation, a handful of people from here went there and exchanged their genes. A handful of people from here went from here to there and exchanged genes. And we actually parameterize all of those things under this model. So what I did, so my postdoc advisor, Jody, he already developed a method called IMA2, which is an MCMC, so Markov chain Monte Carlo based, so simulation based approach towards estimating parameters under that model from genetic data. And so the data here is multi-locus genotype data. So all those reds and blues across the entire genome. And uses MCMC in this case to sample densities sort of across all possible combinations of those trees that I showed you. But the problem with any kind of sort of MCMC method for folks that are familiar with it is that it's slow to converge depending on the size of the data set. And larger data sets also require more time as well as more chains to be able to explore the sort of total posterior density distribution of the parameters. So it makes it really slow. So what I did was, as part of my postdoc work, I built a parallel algorithm for doing more efficient sampling of these methods. So multiple chains on every single processor. You can imagine if you have hundreds of processors, you can basically build hundreds of chains across each one of those processors, samples the entire space of parameters much more effectively and faster. And then you can just collate everything into one and then provide your estimates. So technically, it should be what's called embarrassingly parallel, which means the more processors you throw at it, the better it runs. That's exactly what it does, which is pretty cool. I was very happy to see this. So this was just a simple case study that I did. So in this case, I only used 300 of those genes, distributed essentially across 1 to 20 processors here. So x-axis here is just number of CPUs. And then this is just the total number of iterations that were completed in this case. You can kind of clearly see the more of the processors you throw at it, the algorithm does more iterations, which is pretty cool. This was published a few years ago. So then I wanted to see, hey, can we actually estimate population structure from across these, the Hadza, the Sandawe, and Baka and Yoruba populations. And here's what we found. This was another paper that came out a few years ago, a couple of years ago, where we actually determined, as it turns out, the best model that actually fits the evolutionary history of these 4 populations is one where, again, let me point you to what's going on here. Past is here at the bottom, present day is at the top, so past to present. About 850,000 years ago, which is actually prior to even the generation or the evolution of modern humans, anatomically modern humans, there happened to be some constant sort of ancestral population of all modern day hominid species that split off to form what's called a ghost population. So these are populations that are potentially now extinct. These are non-human hominid species that are potentially now extinct, which split off from the common ancestral population about 850,000 years ago. So it's a very ancient split, even before the evolution of modern humans. And then after that happened, as it turns out, about 50,000 years ago, which is more on the timelines of when humans actually evolved, when humans actually were thriving, anatomically modern humans were thriving. Turns out the Baka population split off around 50,000 years ago from this common ancestral population. And then at somewhere around, that's the time when it split off to form this Yoruba population about 40,000 years ago. And then these Hadza and Sandawe populations split off from each other about 20,000 years ago. So relatively recent. But as it turns out here, the width of these boxes represents the sizes of these populations. So as it turns out, this ghost population, this now potentially extinct population, was very large. But if you look at the size of the Hadza population, it's very small. And this is because of what we call a bottleneck event, where only a handful of individuals from this population essentially went on to establish these populations. So if you look at the genetic composition of all the Hadza peoples, they are highly structured, which means they would look very similar to each other, which is exactly what we saw when we estimated population structure using multi-cluster, for instance. All the Hadza stood out as a very unique population. Now, what was really interesting that we determined, and if you remember right from that figure, there seemed to be lots of gene flow between those populations. And it makes no sense socially because we know anthropologically that these populations have not exchanged genes with each other. So here's what we found. We found that there was actually no significant gene flow between these populations directly, but instead, they all at some point had been exchanging genes with this now potentially extinct population of non-human hominid species, which is pretty cool, okay? And it's Africa. Unfortunately, Africa is also very hot, so climatically it's impossible for fossilized remains to actually be maintained. So for all you know, we'll never know what the species was, because we'll probably never find fossilized remains. Unlike, say, Neanderthals and other species that we've actually discovered fossilized remains in permafrost in Siberia. Yeah, sorry, go ahead.
Do the green lines, I mean, they're distributed at different times on vertical scale.
Correct.
Does that make any, I mean, it looks like the Baka got genes from this ghost population just a few thousand years ago.
Oh, no. Oh, you mean this is not, okay, that's not.
It's not indicating.
No, it's not indicating time. It's just, in this case, just how the figure is. Yeah. It's not actual time. Turns out, actually, time of migration is not identifiable. And so I've been actually developing some other methods to determine when they exchange genes as well between each other.
But the arrow just indicates some percentage of genes.
That's it. Yep. Which is still pretty cool. I mean, 17%, right? That's a large number, which means a large number of these ancestral ghosts actually moved and exchanged genes with the ancestors of the Baka populations. Yeah.
I'm sorry, but you didn't have any fossilized remains or anything. How did you get the DNA from the case?
We didn't get any DNA. So this was another missing data problem, right? So we actually, it's fitting the model essentially. We built a model based on what we sampled here to estimate which model fits this data the best. Turns out this was the model that fitted the best. This is pretty cool, right? So this actually kind of seems to explain why you see so much of gene flow between the sampled populations. Even though they did not exchange genes with each other directly, they actually exchange genes with some who knows what population. Pretty cool. So where do we go from here, right? So we're obviously analyzing only 4 populations in my case study that I showed you. I have an NIH grant right now to be able to study more and more human populations. Again, so these are, again, large-scale genomic data sets, both publicly available as well as data sets that I've generated with my collaborators. And we're going to study across human populations, sort of this presence and absence of these ghost populations across modern human genomes. We know at least 2 ghost populations, Neanderthals and Denisovans. We were actually been able to sequence whole genomes from these fossilized remains that are present in permafrost. But there are potentially, over evolutionary history, we know that there ought to have been multiple such incidents of gene flow from unsampled ghost populations. Yeah.
So you've deduced something about the genome of this ghost population statistics. Is there enough information to compare to actual genomes like
with Denisovans? Yes. Yeah. So those are the only 2 genomes that are actually
right, but can you compare your ghost population
to those? Yes. Yep. Yep. Yeah. So we're reconstructing what this ghost essentially looks like, right? Their genome, and then you can actually compare that with other ghost populations, essentially.
So there's enough information.
There is. Yes. Yep. Yeah. Yeah, and so we're actually much more interested, especially in the context of the sort of this NIH grant, is to understand. There have been some recent studies that have actually shown that these genes that have come from Neanderthals into the ancestors of modern humans in China and some Southeast Asian populations. There was this paper that was published earlier on in the outbreak of the pandemic that showed that individuals that actually carried those specific genes that came from Neanderthals, or I think this was Denisovans, had higher susceptibility to SARS-CoV-2. Okay, so you can kind of think about how we still, all of us humans, have parts of our genome that came from some of these ghost populations, which are now extinct. And statistics, over the years, we've learned, especially if we're looking at Neanderthal and Denisovan DNA, for instance, Central Europeans or people of Central European descent have anywhere between 2 to 3% of their genomes from Neanderthals and Denisovans. East Asians, Polynesians, and Melanesian populations have about 3 to 5% of their genome derived from ancestral ghost populations. We determined that about 2 to 3% or more of native African people's genomes are also derived from some ghost population. So you can kind of see where this is going, right? So if we still retained some parts of this ghost DNA, it begs the question, why, right? And how? Why did we retain some parts of the genome, whereas other parts of the genome were swamped by human DNA? And so we're actually studying sort of functionally, why would some parts of this genome be specifically Neanderthal or specifically derived from one ghost? Does it have any consequences for human health in terms of, hey, just because I have this one particular gene from this ancestral ghost, does it make me much more susceptible to a particular type of adrenal cancer? So our interest is essentially in understanding human health consequences of these gene flow from these ghost populations, and how do we determine them? So that's what we're doing right now on this NIH grant. So we're trying to sort of analyze large-scale genomic locations and understand their functional consequences as to what is actually going on physically at these genes, which makes them be derived or be present in modern human genomes. So with that, I'm going to give plenty of time for questions. Lots of my collaborators, my grad students, Melissa and Jared, who helped me with a lot of this stuff. Funding, you can find me on Twitter, look at my website, but importantly, especially if you're a student listening there or here, I've got a bunch of really good projects, especially working on sort of advancements of these. I want to put a plug for this. I've got 2 fully funded positions in computational sciences as well as in evolutionary biology. Either through the JDP program in EB or through the one in CSRC, as well as 2 fully funded master's positions. So if you want to learn more about some of the projects in my lab, come chat with me, or we can talk more. Okay, with that, I'll stop.
What is up this one?
Yeah.
On the slide that showed the ghosts.
Sure.
What do the numbers mean associated with the arrow?
Oh, those are sort of the degree of gene flow from the ghost.
Okay.
So
not related to percentage of.
No, it's not. Yeah. So this is just how much. Yeah, some of them are pretty huge, right? 30. So it basically.
It's not 30% of it.
No. It's not 30% of the genome. It's just saying that it is higher degree of gene flow compared to this one. That's all. Yeah.
Maybe you already said this, but when did the gene flow happen from the ghost population? Like, do you speculate?
Good question. Yeah. So as it turns out, statistically, at least under this model, it's not identifiable. So estimates of time are not possible under this model. So what we're doing is actually building better models to be able to actually identify what the time of migrations are. So we don't know that answer to that yet. Yeah.
But you do show gene flow to an ancestral population.
We do,
Time's
yes.
bounded.
Yeah, it is bounded between these two, though, right? But this could have happened anywhere in the last 50,000 years, and that's a pretty large time. Yeah.
So the bottom one happens
Somewhere between, it's
800.
a pretty large.
I mean, it's not like it's a scale.
It's not to scale. Yes, please, not to scale. That's why I wanted to. Yeah, I wanted to fit this into the slide. Yeah, definitely not to scale, right? And that's a huge timeline, right? So if you think about that, that's 800,000 years of gene flow potential. So yeah, timing of gene flow has been a real difficult problem to solve, unfortunately. But if you're interested, we can chat about it.
What other, if there are any other mechanisms are there?
Mating is the biggest one, but there are a couple of other ones, actually. That's a great question. So viral transfer is also very rampant across different species. We actually know that a majority of human genomes are actually what are called transposable elements, and they were actually formed by retroviral elements over evolutionary time. A lot of the human genome is comprised of. So it's essentially viruses that enter your body and then get incorporated into your genome and then get passed on to the next generation. And that keeps happening over thousands and thousands of years. So that's another way of transfer of DNA.
Which I guess I probably guess what it's doing then. Is it possible that's not a human, so maybe it's like mosquitoes?
Oh, no, it had to have actually come from, in this case, it had to have come from another species. So it's not, in this case, from a mosquito or a virus, yeah.
That ghost is a homo.
Yes, it is, yep. So this 850,000 is within the time scale of when, within when modern humans evolved. So this time period is when modern humans evolved. So, yeah.
Okay, so just to make sure I'm understanding this correctly.
Sure.
Your proposition is that each of these 4 populations, which were not interacting with each other, were interacting with the ghost population at the time that they were not interacting with each other.
Correct.
But they weren't interacting with each other, you were saying, because of, like geographical considerations of Africa and base right far apart. So like, why would the ghost population not be subject to geographical constraints?
I have no idea. Maybe it's somewhere in the middle, right? So, again, if you look at Africa, right? So, so these populations, especially, they're separated by the Congo rivers and a couple of like huge tropical forests in the middle. So again, I'm not an anthropologist, so I'll leave it to the experts to speculate about that. Yeah, great question. Makes you wonder, right? Where did these populations actually exist? Yeah.
I have a theory. So
Yeah.
The ghosts were fishermen, so they were
Yeah, maybe
like
going up and down.
across
Yeah.
the thing, trading. Could you please stop sharing so that the people on the
What's that?
could you stop sharing so that the
Oh
people
yeah,
on the-
sorry.
see there is any question for you.
Yes, I come up with,
Yeah, go ahead.
yeah, so with the ghost ancestry, well, if we try to project 1,000 years or 10,000 years more, that content of ghost ancestry will disappear eventually. It's like in decay.
Great question. Yes, it is always in decay, but it also stabilizes after a certain point. And if you think about why it's stabilized,
Not in zero, you say stabilized.
stabilized at say 2-5%, but it's always on its way out because we are continually, as humans, as a species, we are still mating and crossing our genes on. And every single generation, some percentage more of human stuff keeps getting added. So that's one thing. But you're also correct in that maybe, let's say, me having Neanderthal DNA, a particular gene, makes me more, I don't know, I can fly and I can mate more or something like that, right? So that we call that as positive natural selection. So maybe it makes me more fit, which means that my offspring will definitely get that DNA, that Neanderthal DNA.
Survive with that. That is my
So that's also possible.
I'm thinking that maybe this is just a statistic, but we can think that that very small percentage of those ancestry basically is a method or a solution of resiliency of humankind, basically.
Very possible. Yep.
We have a surprise of a new virus, and still we have this reserve
Yep.
of weird genetics, which maybe can have a very good solution in order to attack that.
I don't know.
That's
Yeah. So we still have to figure that out. So that's actually one big thing that we're studying in my lab. We're trying to understand functionally, why are only some parts of the human genome from Neanderthal, some parts from Denisovans, some parts from this ancestral ghost, right? Why? What happened physically? Is it because it's causing the individuals to have greater resilience to something? Is it because, I don't know, right? So we have to figure it out.
Because I would think that it's a kind of optimization at the end, why you are trying to keep the best thing
In the genetic material. Yeah, sorry.
It looks like your techniques are only applicable to sexual populations.
In this case, yes. Yep.
So you don't look at.
Yeah, I have really not.
Viruses, forget it, right?
Yeah, I haven't really looked at viruses or even plants for that matter. Plants do crazy things. Plants have, like humans, for instance, have 2 sets of chromosomes, one from mom, one from dad. There are lots of species of plants have multiple sets of chromosomes. That just kind of muddles the data up quite a bit. So yes, in this case, this is only to sexually reproducing species and not so much for anything asexual, or anything different from sexual reproduction. But yeah, no, there's severe dearth of methods for that too, really, because asexual species evolve very differently. Like viruses, for instance, right, evolve very differently. And we still need to understand what's going on, how they evolve as well. And so there's need for development of new methods to be able to understand viral evolution and stuff like that too. And viral genomes are smaller, which makes it much, in my head as a computational scientist, it makes it easier because it's smaller data set. These things are gigantic, so. Somebody had a question on there. I think I saw a raised hand. Go ahead and unmute, and somebody had a question up here on Zoom.
Hi, thanks for the talk. It was very interesting. So my question was kind of related to that previous question, but I was wondering, like, what happens if you have a population that evolves relatively fast? Like, how effective would this method be? Because I would imagine there'd be more gaps in the evolutionary record, so to speak.
So the rate of evolution is one of the things that we actually parameterize as part of the model. And in simplistic terms, we call it a mutation rate.
Yes.
And so on an average, let's say in a human species, we expect, about, one mutation in every million basis is usually what is like the accepted account right now. So at least we can parameterize it, but different species evolve differently. Like you say, like viruses evolve very quickly. Humans, on the other hand, it's much more slower process. So it's all of these estimates that I showed you are all scaled based on the human mutation rate.
I mean, does that make it easier for your method, or does that make it harder if they mutate?
No,
faster?
it's just one parameter, so everything is just kind of scaled by that. So you just multiply everything by the mutation.
So in theory, you could use this for like, if you wanted to track like antibiotic resistance or something like that.
It is not good, only for sexually reproducing species in this case. So yes, you could use it, say, for, I don't know, fast evolving. I'm trying to think.
How about like a pest that's evolving?
Yeah, some pests like Drosophila or like mosquitoes or something like that. Yeah.
Okay, thank you.
Yeah, I also forgot to mention, right? So these are sort of big methods that there are lots of sort of these direct-to-consumer companies like Ancestry.com, 23andMe. There's a whole bunch of those these days that essentially use these kinds of methods. I don't know how many of you have done Ancestry.com or 23andMe. You spit in a cup, you send it off. I showed you that Ancestry Proportion graph, right? The y-axis is what they send you back, right? So they send you back the percentage of your genome that came from one part, percentage of your genome that came from somewhere else. These days, they've actually added some Neanderthal DNA to their database as well. So they'll tell you what percentage of your genome comes from Neanderthals too.
How good are the results?
They're pretty good. I mean, they're incremental though, right? Because the more data they have in their database. So, I think every few months they send an update. So you'll see your numbers change in real time as they get more data. At least that's the first part, right? So this population structure part. The second part of my talk, it's very relevant when we think about not just humans, but when we think about evolution of, say, species that are of conservation concern, or agricultural pests, or domestication history of different crops and things like that. So same kinds of methods are very applicable to a variety of species, which is pretty cool. Hence that's why I have NSF, and I tap into NSF, NIH, and USDA at the same time, because I'm able to sort of apply the same methods to, yeah.
You said to domesticated species, but that's not random.
No, it's not. Yeah, it's not. Yeah, so, but they mate within a subpopulation more randomly.
Yeah, but very selective.
Yeah, it is very selective though, yeah. But you can still track down the population history. Let's say that you isolate a bunch of individuals, you only mate them within each other. So if you look at, say, one of these, right? So the assumption here is that individuals within this population mate randomly with each other, but they don't mate randomly with other populations. That's the idea. So it's the same idea when you have domesticated species. You have a group of crops, let's say, that you selectively breed, or a group of animals that you selectively breed, so they will be more identical genetically to each other compared to others. That's all. So they're structured populations.
Seems like a lot of interesting questions there about how fast you could domesticate or select a certain trait.
Yep.
But random mating isn't going to answer that.
No, it's not. It'll be within random mating within a population. Right? So, yeah, within a group of individuals. Yeah.
So this graph does imply that ghosts were around past 21,000.
No, yeah, that's a good question, actually. So the model assumes that they were around, but the truth is that they probably went extinct at some point. So again, this goes back to the number of models sort of time, increases super exponentially depending on the number of populations, subpopulations. And so if I add an extra time here, that just adds more parameters. And it just started to become a lot much more unidentifiable when I added time of extinction and things like that.
So what am I missing? I mean, the 21K years is sort of lined up with when the sun Dolly and the Hansa split off. and they have different gene flow from the ghosts, how did that happen?
Right,
If
so.
they have a common ancestor.
Yeah, in this case, it does actually suggest, yeah, you're right. So it does suggest that these ghosts actually exist past that, because they have to have exchange genes for the similarity.
With that note, we're gonna thank our speaker one more time.
Thank you.